The Van Bekkum is EBMT's most prestigious award and offers attendees an insight into the most exciting developments in the field. Here to discuss her award-winning paper looking at the use of cyclophosphamide in graft versus host disease, I'm joined by Leslie Kean. Leslie, thank you so much for joining us.
Thanks for having me.
Many congratulations on the award. Let's start by talking about where the field is in the use of cyclophosphamide today and what were you hoping to find with the study?
Thank you. I'm so honored to present this work on behalf of the Bone Marrow Transplant Clinical Trials Network, or the Bmt-Ctn, 1703 and 1801 study. This study, which was published in the New England Journal in 2023, sought to determine whether post-transplant cyclophosphamide could potentially be a new standard for the field for GVHD prophylaxis. And when looking at a combined endpoint, the combined survival with chronic GVHD, post-transplant cyclophosphamide was the clear winner compared to more standard prophylaxis with tacrolimus and methotrexate. But what this study sought to do was to understand the underlying biology of that result and also to determine whether there were any trade-offs with cyclophosphamide that the field should be aware of.
So the data you gathered for the study was via T-cell receptor or TCR immunosequencing. What was special about this approach and what's the kind of data that you gathered able to tell us?
Yes, so we combined TCR sequencing with multi-parameter flow cytometry and also single-cell RNA sequencing. But what the TCR sequencing allowed us to do is to evaluate the T cells that reconstitute after transplant at a level of detail that was unprecedented in the field. We were able to profile almost 200 million T cells from these patients on this trial, and it allowed us to draw conclusions that simply were not possible to determine before.
So what were those conclusions? What were the key takeaways, and what does that mean for the future of patient treatment and care?
What we have found is what we think is the reason that post-transplant cyclophosphamide has a positive benefit on chronic GVHD, but may have some risks with infection. We find that the diversity of T cells that reconstitute after transplant in the presence of post-transplant cyclophosphamide is much lower than what the diversity of the T cells that reconstitute after tacrolimus and methotrexate. And we think that this diversity is lower because there are fewer naive T cells. And what we have found is that we can establish a threshold above which, for naive T cells, above which patients are at significant risk for chronic GVHD, and below which occurs with post-transplant cyclophosphamide, where they're much more protected. The problem is that there's also probably a trade-off here. And when you have fewer naive T cells, fewer TCR singletons, you are also potentially at higher risk for infections, including for COVID-19.
And so how do clinicians balance that trade-off then?
I think the most important thing that clinicians need is to be aware of the trade-offs. We cannot anymore in this field say that one prophylaxis regimen or another is quote unquote the best. We have to think about each individual patient and what they need. Are they more at risk for chronic GVHD? Are they more at risk for infection or toxicity? And make targeted decisions based on our targeted science that we now understand with these prophylaxis regimens.
How has the EBMT impacted your research and clinical practice?
The EBMT is one of the cornerstone organizations in our field internationally. I am a physician that practices in the US, and I'm very active with the American foundations and organizations. And we work hand in hand with the EBMT to make sure that all patients are provided transplants that will be most successful and cure their underlying disease.
I understand that your research is a first in TCR sequencing studies. Tell us about that and what it means to you to be recognized by your peers.
Yes, so TCR sequencing has been an established method for evaluating post-transplant reconstitution, but there's never been a study that has evaluated TCR sequencing and its role in evaluating post-transplant immune reconstitution to the depth and the breadth of this study. So it has been an incredible opportunity and honor to be able to look at patients this deeply. And because we were able to do that, we were able to draw conclusions that simply were not possible before. So I would just say that it's been an honor to be able to do this work and to really look at our patients from this new viewpoint.
And just to finish, what's next, Leslie? Is there more work to be done in this area or new topics that you'll be looking at?
There's much more work to be done in this area. This is only the first study to really look at TCR reconstitution and understand at a molecular and a cellular level what might be underlying both efficacy of transplantation and toxicity. And I think that's where the field is. We are at this time during transplant that we have so much success, but that we have to start looking at trade-offs and understanding for each targeted therapy that we give our patients, what are the risks and the benefits of that therapy.
Leslie, thank you so much for dropping by to speak with us, and many congratulations again.
Thank you so much.